I'm Adi Moore, co-founder, CEO, and CSO of Chemomab Therapeutics, and it's a pleasure to be presenting at ROTH MKM conference. Please note I will be making forward-looking statements, so please refer to the risk factors in our SEC filings. Chemomab is a clinical stage company developing CM101, a first-in-class monoclonal antibody that neutralizes CCL24, a unique dual-acting target central to the biology of fibrosis and inflammation. CM101 has demonstrated disease-modifying potential in multiple fibro-inflammatory disorders. We are targeting rare diseases with large commercial potential and high unmet need, initially primary sclerosing cholangitis, PSC, and systemic sclerosis, SSC. We believe the CM101 program is substantially de-risked by the extensive preclinical and early clinical data we have assembled over the years, including 4 clinical trials demonstrating both safety and activity with consistent positive biomarker responses. We recently extended our cash runway through the end of 2024, assuring we have the resources on hand to reach our PSC top-line readout in the second half of 2024, which is a potential major catalyst event for Chemomab. Based on the very comprehensive preclinical and clinical data showing CM101's strong interference in inflammation and fibrosis in the liver, in the skin, and in the lung, we consider CM101 to serve as a pipeline in a drug, and we see great potential in its disease-modifying activities across multiple indications. Our lead indication is PSC, a rare liver fibrotic disease with no approved drugs. CM101 is currently being evaluated in a phase 2 clinical trial, and we expect to report top-line results from this trial in the second half of next year. A second indication, systemic sclerosis, it's another orphan indication lacking any effective therapies that involves fibrosis in the skin, in the lung, as well as additional internal organs. The CM101 SSC program is currently phase 2 ready. And earlier this year, we reported positive results from our phase 2 liver fibrosis trial in NASH patients. Although we are currently not pursuing NASH as an indication, we do believe that the promising results from this study are de-risking and very relevant to the PSC study, as well as other fibrotic diseases. So who is CCL24? It's a soluble protein that belongs to a subfamily of cytokines that was found to play a dual central role in promoting directly both inflammation and fibrosis. The ability of CCL24 to induce these two pathways simultaneously and directly is very unique for a target, and that's led to the generation of CM101, a first-in-class antibody that binds CCL24 and neutralizes these effects. And indeed, we're able to demonstrate that CM101 holds a differentiated dual mechanism of action, being able to break the vicious cycle that eventually leads to fibrosis. Over the years, we have generated a very comprehensive preclinical package, including more than 12 different animal models, as well as multiple in vitro and ex vivo studies. The results from these studies, which include liver, skin, and lung fibrosis-related models, provided substantial evidence of CM101's anti-fibrotic and anti-inflammatory activity, and definitely encouraged us to advance towards clinical trials. Our lead program is primary sclerosing cholangitis. PSC is a rare disease characterized by obstructive damage to the bile duct that eventually leads to inflammation, fibrosis, end-stage liver disease, and a high need for liver transplantation and occurrence of liver cancer. It's a terrible disease that affects mainly men and lacks any FDA-approved drugs. Unfortunately, the only treatment with curative potential is liver transplant, and even there we can see high recurrence of the disease. In terms of the market, it's a serious unmet medical need with approximately 80,000 patients estimated in the 7 major markets, and a commercial opportunity that is estimated beyond $1 billion. There are several ongoing clinical trials in PSC. The vast majority of the drugs being tested are targeting bile acid metabolism and not disease-modifying activities like CM101. A phase 2 study testing an anti-fibrotic integrin inhibitor by Pliant was recently reported, the phase 2 data showing some encouraging results in the field. Giving CM101 mechanism of action that includes not only anti-fibrotic effects, but also direct anti-inflammatory effects, we believe that CM101 may have significant added value as well as clear differentiating properties. As mentioned, we have conducted multiple preclinical models over the years. Here is one example, the TAA liver fibrosis model in rats. As you can see in the livers on the left, TAA alone resulted in a very significant fibrotic damage, while treatment with CM101 in the middle resulted in livers that looks very similar to the healthy one. Based on multiple positive preclinical studies similar to the one presented here, as well as early clinical data, Chemomab launched its phase 2 clinical trial in primary sclerosing cholangitis. Here's the design of our ongoing phase 2 study in PSC. It's a randomized placebo-controlled study where a total of 68 patients received 10 or 20 mg per kg versus placebo every 3 weeks over 15 weeks period, and then had the option to continue into a 33-week open label extension where all participants will receive the drug. The primary objective of the trial is to evaluate the safety and tolerability of CM101, and the secondary outcome measures includes evaluation of a range of relevant biomarkers, ELF, FibroScan, PRO-C3, ALP, and others. As mentioned, we are anticipating top-line data in the second half of 2024. We believe that this study is well designed to evaluate both the safety and potential activity of CM101 in patients with PSC, and positive data should support continuing into a phase 3 for registration. In addition to the ongoing PSC program, we reported earlier this year positive data from a phase 2A liver fibrosis study in NASH patients. Given the similarity in the biomarkers that are being evaluated in NASH and PSC, we do believe that the data obtained from the liver fibrosis study that I will now present may serve as a translational bridge to what we might expect to see with PSC, where we are treating patients, larger groups of patients with higher doses of CM101 and for a longer duration. This trial was a randomized placebo-controlled trial that evaluated 8 repeated doses of CM101 administered subcutaneously at a dose of 5 mg per kg over 16 weeks in 23 NASH patients with liver fibrosis. The primary objective to assess safety and tolerability was met, and CM101 appeared to be safe in this study. Most adverse events were mild and no drug-related serious adverse events were observed. To evaluate the activity of CM101, we measured a variety of biomarkers that are known to be relevant to inflammation and fibrosis and are commonly used to assess the stage of liver fibrosis, including ELF, different collagens, and others. Using a responder analysis, we demonstrated that higher percentage of patients in the CM101 treated group showed improvement in these biomarkers, as you can see in the blue bars, and this is compared to the placebo group indicated by the red bars. In addition, we evaluated the biomarker improvement in patients who were defined as multiple responders. Those are patients who improved in at least 3 different biomarkers. Importantly, the data showed that almost 60% of the CM101 group responded in at least 3 biomarkers compared to 0 in the placebo group. These findings further show the broad effect of CM101 on multiple biomarkers and suggest that it's inhibiting core pathological processes that eventually lead to fibrosis. Based on 4 completed clinical trials to date, we see that CM101 appeared safe and well tolerated with consistent PK profiles and no signs of immunogenicity. From the activity point of view, CM101 was shown to improve both inflammatory and fibrotic biomarkers in several patients' population that involve both liver and lung damage. These biomarkers that are also very associated with primary sclerosing cholangitis as well will be tested in our upcoming study readout and lead the way towards a phase 3 program. In summary, with its unique dual antifibrotic and anti-inflammatory mechanism, CM101 represents a potential breakthrough with application to multiple rare diseases with high unmet need. Given its unique targeted properties, there are multiple opportunities for expanding CM101 to other fibro-inflammatory indications going forward, which we definitely plan to explore in the future. Finally, Chemomab has sufficient cash on hand to take the company through its PSC phase 2 readout, a potential major catalyst for the company. I'd like to thank you all for your time and attention.